Okay, all of you listen to me very carefully. Mostly try to use this particular PPTs only, whichever I am giving you right now in this particular classes. Okay, so don't take the printout of photosynthesis until I complete the concept of the photosynthesis, because it will take 2 to 3 classes, because we have to study the light reactions also. We have to study the dark reactions also. So I will be keep updating the PowerPoint. So until I complete the PowerPoint, complete the concept, don't take the printout. Once I complete it, then you people take the printout. Okay. Okay. Because I have added a lot of extra information, which every one of you has to remember compulsory. Okay. Good morning. Good morning, everyone. Okay. From tomorrow onwards, please be on sharp time. Okay. Okay. Every one of you listen to me very correctly, and do not disturb me for the period of 30 minutes. I will explain each and everything from very basic to the very depth. Now, what we have to study in the chapter of plant physiology. I told to all of you friends in the last class also, from the plant physiology chapter, friends, you can expect 10 to 15 questions very easily, either in the JL paper or in the DL paper. Why? Because plant physiology is the chapter where you can get the questions purely conceptual. You can get the analytical questions, you can get the matching questions, you can get the statement questions. You can expect different type of questions from the plant physiology chapter. Okay, and according to our syllabus, what we have to read, good morning, read in the chapter of plant physiology, we have to read photosynthesis, respiration and photorespiration, nitrogen metabolism, plant hormones, sensory photobiology, transport in plants. See, secondary metabolites, we will see it. I will once again check the previous year papers, but it is one of the most important topic that we have to discuss compulsory. Then, friends, followed by the stress physiology. So these are all the various topics that we have to study in the plant physiology. And remember, every one of you, from every single topic, you will expect the question very, very surely. Okay, now how to read the entire chapter? All the students listen to me very correctly. Suppose example, I have finished photosynthesis today. Example, I will provide you the PDF immediately. So what you have to do? You have already PDFs. Immediately, you keep the PPT with you, and whatever the PDF that I will provide, in that PDF, read the entire photosynthesis concept. And in that PDF only, there are hundreds of practice questions are there. Even I will also solve the practice questions. So that is how you have to read. Don't think that if you listen the lesson, it is sufficient. Of course, see, my lessons will help you 90%. 90% of the question will come from whatever I am speaking in the class. Remaining 10% to get the enough knowledge, it is very important. You have to read the book compulsory. Okay, especially the extra information that what I teach in the class. Okay, now let's start. Photosynthesis. Don't disturb me for half an hour. Just mute your mics and listen to me carefully and answer all my questions. Okay, listen friends. What is photosynthesis? From 10th standard, we are reading the definition. Photo, light, synthesis, to produce, to make something. So what actually plant will make, friends? Plant will actually make its food. So what is the food for the plant? Of course, sugars. So now, in simple words, in a very simple words, I can tell you that the reduction of the carbon, what friends? Reduction of the carbon into sugar. That is what exactly we can call photosynthesis. So what are the various raw materials required for the photosynthesis? There is a requirement of the sunlight, which is coming from the sun, and requirement of water coming from the soil, and requirement of the carbon dioxide coming from where, friends? Atmosphere. So friends, this particular carbon dioxide, with the help of a chlorophyll molecule, the plant is reducing the carbon into sugar. All the students try to understand. So plant is preparing its food. Whenever plant is preparing its food, plant is releasing what? Plant is releasing oxygen as what product? Oxygen as the byproduct. Oxygen as the end product. Oxygen as the waste product. Every student remember for the exam, no plant or none of the plant on this planet Earth never ever release the oxygen purposely. Every plant release the oxygen as a end product or a waste product. That is the oxygen what we are actually breathing. Never confuse and never forget that. And you studied this particular equation in the 10th standard. Remember the equation as it is: 6 carbon dioxide, 6 water, 1 sugar molecule, 6 oxygens. Just remember 6616. Now friends, 80 to 9, please mute your mics. Do not create any kind of a disturbance. 80 to 90% of the questions in our competitive exam, friends, will come from locations, which means where does light reactions takes place, where does the dark reactions takes place, where the electron transport chain is present, where the photosystems are present, where the electron acceptor complexes are present, where the breakdown of water occur, where the ATP synthesis will takes place, where the NADPH synthesis will takes place. So friends, 90% of the questions comes from locations. So that's why every student compulsory has to remember the locations, not only in this chapter, every single chapter. Now listen every one of you very carefully. Where does the photosynthesis occur? Every student know that photosynthesis occur in a specialized organelle called as chloroplast. So listen to me. I will cover maximum amount of the information. By that you no need to read anything extra. Listen to me. So if you look into the structure of a chloroplast, you can see the diagram very clearly, friends. Chloroplast is a double membrane organelle, consists of a inner membrane, consists of a outer membrane. The space between the inner and outer membrane, we called it as the intermembrane space. And friends, if you look into the chloroplast inside, if you look into the inside the chloroplast, you can see here there is a flattened vesicle is present. You can see this. What do you call this particular flattened vesicle? You call this particular flattened vesicle as thylakoid. But friends, what is the interesting point? Inside the chloroplast, there is a no single thylakoid. Friends, inside the chloroplast, there are many groups of thylakoids are present. You can see here, thylakoids are arranged one on one, one on one, one on one, like a group. So friends, remember, the group of thylakoids, what do we call it as? We call it as granum. Every student remember the point. Inside the chloroplast, there will be a no arrangement of the single thylakoids. Every time, friends, thylakoids are arranged in the form of a group only. Never forget this particular statement. Now, friends, one group of thylakoid can be connected to the another group of the thylakoid. How it will be connected, friends? By a thin thread-like structure. So, friends, what is that thin thread-like structure called as lamella? Not to confuse, friends. So, this is one group of thylakoids. This is another group of thylakoids. These two can be connected with each other by a thread-like structure. This is called lamella, not to get confused. Once again, quickly recall, chloroplast is a double membrane organelle. Focus everyone, whomever joined right now, mute your mics. It consists of outer membrane, consists of inner membrane. In between the space is called intermembrane. Inside the chloroplast, the flattened vesicle is called as thylakoid. And what I'm trying to say to all of you, every time thylakoids are arranged in a group, that group of the thylakoids only we can call granum. So one granum can be associated with the another granum with the help of a thread we can call lamella. And friends, look into the picture. Inside the chloroplast, All the students listen. Inside the chloroplast, fluid filled structure. What is the definition I am giving you? I am giving you a definition. Fluid filled structure. You can also call it as matrix. You can also call it as a cytoplasm. There are many number of names are there. Fluid filled structure present inside the chloroplast, that is called stroma. Never forget that. And we all studied many number of times. Chloroplast is what kind of a organelle? Chloroplast is a semi-autonomous organelle. What organelle? Semi-autonomous or self-replicating, which means, friends, it can synthesize proteins on its own. So, friends, if the chloroplast want to synthesize the proteins on its own, what does it require? It requires its own DNA. Every student commonly know that. What is the shape of the DNA that is present inside the chloroplast? It is the circular DNA. All the students remember. You also know very well what is the most important protein synthesized by the chloroplast DNA. It is called rubiscoprotein. I told to all of you many number of times, the most abundant protein on this planet Earth is the rubisco only, which is coded by the chloroplast DNA. You also know very well what kind of ribosomes are present, friends. 70S ribosomes are present inside the chloroplast. All the students look into the diagram, you can see here there is a large starch granules are present and small lipid droplets are present. So every student remember my words. Chloroplast always store abundant amount of the starch when compared to the lipids. I am not saying that chloroplast never store the lipids. Chloroplast also stores the lipids. There is a no doubt about it. But very less amount of the lipid it will store, but more amount of the starch it will store. Because understand, inside the chloroplast, abundant amount of the starch granules are present. Not to get confused with this particular point. So friends, listen. All of you listen. Right now, I have explained you the structure of the chloroplast. The same structure you have studied in the intermediate level, degree level, BSc level, MSc level also. But friends, is there any necessity to study the inner view? What do you mean by the word inner view here? Microscopic structure of the chloroplast. Is it necessary to study? Friends, it is compulsory, it is mandatory to study the inner view. What is the reason? Because friends, inside the chloroplast, different reactions, listen my statements all of you, different reactions will takes place in a different areas. That is the reason you have to study every microscopic structure of the chloroplast. All the students listen. This is what do you call, friends? Thylakoid, a stack of thylakoids or a group of thylakoids, that is called granum. Correct na? All the students please listen. This is called what, friends? A thylakoid membrane. Thylakoid membrane, widely called as lamellae. Never ever confuse. Friends, this is how the thylakoid plasma membrane looks like. It's a very important, you have to know it. This is how the thylakoid lamellae or the plasma membrane looks like. Most of the students will get confused between lamella and lamellae. Every one of you remember, thylakoid membrane is called as lamellae. There is a addition of E here. What is lamella? One group of thylakoids can be joined with another group of thylakoids. That is called lamella, not to get confused with that point. And friends, inside the thylakoid, so you understand the membrane, and inside the thylakoid, you can see, right? This is called lumen. My friends, remember everyone, I am not talking about not only thylakoid, any cell organelle. Remember everyone, any cell organelle, inner space, what do we call it, that inner space? Lumen. So lumen means inner space for any organelle. So in order to understand this, we have to study every single area of the chloroplast compulsory. Now I will explain you why. So friends, entire photosynthesis you can understand in this particular slide. So I'm requesting every student to try to understand and focus from this particular slide. And till the end of the lesson, never forget what is the goal of the photosynthesis. Never forget. What is the ultimate goal of a plant body? The plant has to reduce the carbon into what? Glucose. This is the ultimate goal of the plant. Ultimate goal, reduction of the carbon into glucose. But the point is, friends, if the plant want to reduce the carbon into glucose, it is not easy process. What the plant require, here only I am telling you, if the plant want to reduce the carbon into glucose, light is not important. What is important, friends? Energy is important. If plant contain energy, it is enough. Whether it is the daytime, whether it is the night time, doesn't matter. Plant immediately reduce the carbon into glucose. What the plant only require? Energy, not the light. But what is the interesting point? If the plant wants to produce the energy, what the plant require? Plant compulsory require light. Without light, no plant on this planet Earth never ever produce any kind of energy. So that is the reason, friends, photosynthesis broadly categorized into light reactions and dark reactions. Remember, every students, light reactions means the reaction always takes only in the presence of light. So here itself you're understanding light reactions only occurring in the daytime because the sunlight is only present in the daytime. Never ever forget what is the ultimate goal of the light reactions? Production of ATP, production of NADPH. That is the ultimate goal. What is ATP? Adenosine triphosphate, which is the energy equivalent. Every student know. And I want every student has to remember this. NADPH, we studied in the biochemistry, nicotinamide adenine dinucleotide phosphate. NADPH. My friends, this is the most important statement every student has to remember. NADPH can act as a reducing equivalent. I will show you what kind of questions they will ask you. Sometimes, friends, instead of using the word reducing equivalent in the exam, they will say that NADPH can act as a electron carrier, can act as a electron transporter. That is also absolutely true statement. It can act as reducing equivalent. It can act as electron carrier also. And at the same time, it will provide energy also. But friends, NADPH never provide energy directly. Who will provide the energy directly? ATP will provide the energy directly. Now listen, everyone. So what is dark reactions? So in the light reaction, in the presence of light, plant produced energy. Now friends, plant is using that energy to, by using that energy, what it is doing? It is reducing the carbon into glucose. So that is the reason every student remember. This is also statement asked in the exam. Dark reactions are called as reduction reactions. Light reactions are called as oxidation reactions. It's a very important statement because breaking down occur here and we are reducing the carbon into glucose here. Never ever confuse with this particular point. Always remember. Now, so when the light reactions will be taking place, day or night, undoubtedly light reactions will take place only in the day. And friends, where the dark reactions takes place, it is, see, once again I am telling you, friends, dark reactions doesn't mean that they are occurring in the dark. Dark reactions means they are occurring either day or night. It is light independent reaction. What do you mean by that? Whether the light is present, whether the light is absent, it doesn't matter. Because you have to think with common sense. Plant prepare the food daytime also. Plant prepare the food nighttime also. So dark reactions occur every time. Light reactions occur only daytime. Okay, so now let me explain you 15 minutes. After 15 minutes, I will solve all your doubts. Listen to me very, very carefully from now onwards. So first, our ultimate goal of photosynthesis is what? Ultimate goal is reduction of the carbon into glucose. But friends, unfortunately, plant can't reduce the carbon directly into glucose. First, it has to produce energy compulsory. Without energy, nothing will happen. So how does it produce the energy? In the presence of light. Now let's see how. Light reactions, also called as light dependent reaction. And 2 to 3 times the asked question in the exam, where is the site of light reactions? Where is the site of light reactions? It is taking place on the thylakoid lamellae. It is also called as what, friends? It is also called as thylakoid membrane. Now you all people has to ask me a question. Why? Why the light reactions are taking place only on the thylakoid lamellae? Sir, why it is not taking place in any other area in the chloroplast? Why it is taking place only here? That is a valid doubt. The reason is that on the thylakoid membrane only everything is present. You can see here, photosystem 1 is present, photosystem 2 is present, ATP synthase is present, all the electron acceptors are present. That is the reason the photosynthesis light reactions are only taking place on the lamellae. We will discuss how. Now, friends, you tell me, what is the ultimate goal of the light reactions we were studying? Every student has to understand me. Ultimate goal of the light reaction is production of energy. Now, you all people know very well how the energy come to the planet Earth. Energy will come to the planet Earth from the sun. Who is going to absorb that particular light from the sun? Undoubtedly a pigment, right? Most of the students don't know the classification of the pigments. It is very important to understand the classification of the pigments. Friends, all the pigments present in the plant, it is broadly categorized into two types. One is called principal pigment. Sometimes instead of calling principal pigment, they called reaction center. That is also same. You have to remember. Another one is called what, friends? Accessory pigment. Sometimes instead of calling accessory pigment, they called antenna pigment. Anything is same. Now, what are the example of principal pigment? In the photosynthesis is a type of a topic. 100% of the concept to be understand. 100% of the concept to be remember. It's a very calculative topic. So you have to be very cautious while you are learning it. So principal pigments are chlorophyll A, bacteriochlorophyll A, chlorobium chlorophyll. Now, friends, every student remember my words. They can also ask the question in the exam. Which of the following, remember everyone, which of the following pigment is most abundant in almost, please turn off your audios and videos. Do not interrupt me. I told you multiple times. Please maintain discipline in the classroom and do not disturb me while I'm speaking something. Principal pigments or a reaction center. So what I told you, they are chlorophyll A, bacteriochlorophyll A, chlorobium chlorophyll. Inside the green plant, I mean to say, friends, 90% of the higher green plants, they contain chlorophyll A only. Chlorophyll B, chlorophyll C, chlorophyll D, they never ever present in the higher plants. They will only present in the lower organisms such as the green algae. Always remember, because friends, from here only you can expect many number of questions. I will show you. Okay, now you listen, friends. Now you have to ask me, sir, what is the difference between chlorophyll A, bacteriochlorophyll A, chlorobium chlorophyll? I will give you the very simple definition. All the students understand me. Be active. Modified chlorophyll A. Listen my statements. Modified chlorophyll A, which is present in the purple bacteria. It is called bacteriochlorophyll A. You can ask me, sir, what do you mean by the word modification? Structurally, they got modified. I will show you in the next slide. What do you mean by the word structural modification? Now, next one, bacteriochlorophyll A, same. Modified chlorophyll A present in the purple bacteria called as bacteriochlorophyll A. Modified chlorophyll A present in the green sulfur bacteria, it is called chlorobium chlorophyll. So friends, actually these two are chlorophyll A only, but they got structurally modified. I will show you how they got modified. Now listen, what are accessory pigments or antenna pigments? Chlorophyll B, C, D. They are not at all present in the higher plants. They are present in the lower organisms such as green algae. What are carotenoids? Carotenoids are of two types. First type of carotenoids are called carotenes. Every student remember, carotenes, friends, carotenes contain just carbon, hydrogen, nothing else. Only carbon, only hydrogen. What are xanthophylls? Along with the carbon, hydrogen, it also contain oxygen. That is called xanthophyll. What is the example for the carotenes? Beta carotene. I will tell you in the next slide. You know, right? Beta carotene is the pigment which gives the orange color to the carrot. Lycopene, which is giving red color to the tomato. These two are what? Examples of carotenes. And what is the example of xanthophyll? Example of the xanthophyll is the lutein. Friends, it is very important to remember the examples. I will ask you in the tomorrow class. Now, next one, phycobilins. Where they are present in the cyanobacteria and rhodophyta. Example, phycoerythrin, phycocyanin, yellow phycocyanin. In this particular slide, nothing to explain. You have to remember all compulsory for the exam. But friends, let me tell you one very interesting statement here. This is also very good question for the exam. What is the question? We are having chlorophylls, we are having carotenoids, we are having phycobilins. I want every student has to answer my question. Every one of you type the answer. Among the chlorophyll, carotenoid, phycobilin, which pigment is the water soluble? I want every student has to answer quickly. Type the answer. Every one of you, right or wrong, type it. Which of the pigment is a water soluble among these three? All the students keep the point in your mind. Only phycobilins are the water soluble. Friends, chlorophyll, carotenoid, these two are lipid soluble in nature. 90% of the students will think that chlorophylls are the water soluble. That is absolutely wrong statement, friends. I will show you right now. Chlorophyll is the lipid soluble. Carotenoid is also lipid soluble. Only the phycobilin is the water soluble. Now, every student, let me ask you another very, very good question. This is also a very good logical question. Recently asked a question in the NEET level exam also. You all people studied that chlorophyll A shows bright or blue-green color. Friends, even you can get the same type of a question in the matching. Which pigment show which color? Chlorophyll B shows yellow-green color. Xanthophyll shows yellow color. Carotene shows yellow to yellow-orange color. But where they will show? That's a very good question. Friends, every student, please remember the name of the technique. Whenever we use a technique called paper chromatography, botany people also done compulsory in your BSc and MSc experiments in the biochemistry chapter. Paper chromatography. So friends, in the paper chromatography, there is a paper is present. That paper only called chromatogram. In the chromatogram, when you treat this particular chlorophyll, they will show all these colors. So you no need to remember about the experiment, but remember the name of the technique called paper chromatography. That technique also they will ask in the question directly. Now, every student listen my words correctly. Now, every one of you understand what are the type of photosynthetic pigments. Chlorophyll, carotenoid, phycobilins. Most of the students, they just read the types. They never read the structure. Reading the structure is extremely important. I never encourage any one of you to remember the structure. It is not required. I encourage every one of you to understand the structure. If you understand the structure, question is very easy. First, let's talk about chlorophyll. chlorophyll having a head, chlorophyll having a tail. Friends, what is the head of the chlorophyll is called as? Every student remember, the head of the chlorophyll is called as porphyrin head. The tail of the chlorophyll is called as a phytol chain. Next time when you become a JL or when you are teaching a NEET level, you have to explain compulsory about the head and tail. Now you listen, this is called porphyrin head. All the students listen. Now how can I define the porphyrin head? Tell me. Here you are all able to see A, B, C, D. Friends, you know what it is? It is called pyrrole. What it is? Pyrrole ring. How many pyrroles? 1, 2, 3, 4. So you have to remember my definition. How many pyrroles? 4. A tetrapyrrole ring. What, friends? A tetrapyrrole ring come close together with the help of nitrogen and surrounding central metal atom magnesium. Once again, I'm giving you the definition of the porphyrin ring. Tetrapyrrole come close together with the help of nitrogen and surrounding the central metal atom magnesium. That is called porphyrin ring. What is the phytol chain? Very simple definition. A long hydrocarbon chain. That's all. That is called phytol chain. Just now, I have explained it to all of you. Friends, chlorophyll is a lipid soluble. Why? Because of this phytol chain only. Because of phytol chain only, the chlorophyll is the lipid soluble. And every student quickly answer my question. What is the function of magnesium inside the head? Every one of you. Every one of you answer. What is the function of the magnesium in the head? Remember everyone, magnesium can act like a chromophore. This is also a question you can expect. See friends, we are saying that chlorophyll molecule absorb the light. How chlorophyll molecule absorb the light? With the help of magnesium, chromo, color, 4, which can absorb the color. Which can absorb the color. Whichever is absorbing the color, that is only we can call chromophore. Never get confused with that point. Now friends, we all people just now in the previous slide, we studied that chlorophyll A, chlorophyll B, and we studied bacteria chlorophyll A. Now you only tell me every chlorophyll having same head, every chlorophyll having same tail. Now the question is, how can you differentiate one chlorophyll molecule with the another chlorophyll molecule? How is that possible? It is possible only with the help of a side chain. Never forget friends, chlorophyll A having the side chain of methyl group. Chlorophyll B having the side chain of aldehyde group. Bacteriochlorophyll having some other group. So friends, one chlorophyll molecule can be differentiated from the another chlorophyll molecule with the help of side chain. That also you can expect in the form of a statement. Always remember. So every student understand structure of a chlorophyll. Now every one of you understand the structure of a carotenoid. So remember everyone, carotenoid also called as what structure? Isoprene structure. What is that friends? Isoprene. How can I define the isoprene? Every one of you remember. Isoprene means it is a 5 carbon containing compound. How many? It's a 5 carbon containing compound with the alternate single bond, double bond, single bond, double bond, single bond, double bond, single bond, double bond. Such a simple structure. 5 carbon containing compound with the alternate single bond, double bond. That is only we can call the isoprene structure of the carotenoids. Now, what is the structure of a bilin pigments? Bilin pigments also called as a tetrapyrrole only. So friends, just now where we studied tetrapyrrole, we studied the tetrapyrrole here. Here what is the tetrapyrrole? Circular. But friends, here tetrapyrrole is linear. Linear means straight chain. 1, 2, 3, 4 pyrroles. 4 pyrroles arranged straight. So friends, bilin pigments are linear tetrapyrrole, carotenoids are isoprene units, and chlorophyll contain porphyrin head and phytol chain. These are all the structures of the chlorophyll molecules and other pigments also. Now, if I ask you one single question to everyone, what is the function of every pigment in the plant body? Every one of you give a straightforward answer that all the pigments are required for the absorption of light. That is the answer everyone will give. But the question is that if every pigment is absorbing the light, why the plant require all the pigments? Any one pigment is sufficient, right? Because every pigment is showing same function only. But friends, every pigment absorb the light at a different wavelength. So none of the pigment never ever absorb the light at the same wavelength. So that's why every plant require all the pigments. Now every student very quickly answer my question. Among these three pigments, which pigment will help the plant from the high intensity of sunlight? Or which pigment protect the plant from photooxidative damage? Every one of you. Which pigment is protect the plant from the high intensity of sunlight? All the students remember, chlorophyll never protect the plant from sunlight, excess sunlight. You tell me friends, when we expose to the sunlight, we will run very fast because we can escape. Do you think plant can escape? Plant never escape. So plant has to protect compulsory. How it will protect? With the help of carotenoids. Such an important statement it is. Good. Now listen. Now friends, again 90% of the people will get a lot of confusion between the absorption spectra and action spectra. Okay, every student has to listen to me correctly. I will explain the photosynthesis and other topics in such a way that if any conceptual question will come, every student should be in a position to answer just like that. So you have to focus on the concept very correctly. So there are two type of spectra are there. Absorption spectra, action spectra. I will give you a very simple definition. What is absorption spectra? Absorption spectra, it is always with respect to the pigment, only with respect to the pigment. Action spectra, it is always talk about process. Always it is talking about a process. So how can I define the absorption spectra? At which particular wavelength, all the students try to understand me, at which particular wavelength, what pigment absorbs the what light? Once again, remember the definition. At which particular wavelength, what color absorbed by pigment? That is called absorption spectra. What is action spectra? At which particular wavelength a specific process occur in the plant. I will give the examples to both. All the students follow the graph correctly. On the x-axis, we have taken wavelength of light. On the y-axis, we have taken absorption of light absorbed. You are all able to see in the graph. I have taken three pigments, friends. I have taken chlorophyll A, I have taken chlorophyll B, I have taken carotenoids. All the students look into the graph. Chlorophyll A absorb. See the graph. The graph of chlorophyll A is very high till here. So friends, chlorophyll A absorb dark blue color. Chlorophyll A absorb light blue color. Now you tell me, does the chlorophyll A is absorbing the green color? Never, ever. It always reflect. So every student remember the definition. Remember the definition. Whichever the color, see, I am a pigment. Whichever the color I absorb, I never look like that color. But whichever the color I will reflect, I will oppose, I will obviously look in that color. So friends, what I'm trying to say, chlorophyll A never absorb green color. it reflect, it oppose the green color. That's why all the plants are green in color, because chlorophyll A reflecting the green color. And you know, you look into the yellow color one, what is that? Carotenoids. So you see here, carotenoid absorbing dark blue color, light blue color, green color. But carotenoid is not absorbing yellow color, not absorbing red color, not absorbing orange color. You know very well, carrot is what color? Orange color. Why? Because inside the carrot, carotene is present. It is reflecting the orange color. So carrot is orange color. Tomato is red in color. Capsicum green in color. They contains what? They contain carotenoids. They keep on reflecting a particular color. Whichever the color they reflect, they look like in that color only. So that is what? Absorption spectra. Never confuse and never forget it. Next one. Action spectra. I want everyone to be active. I will take up to 6:15. Okay, be active. Action spectra. What I'm telling about the action spectra? Action spectra always talk about process. Means friends, at which particular wavelength a particular process is occurring. You take any example for the as a process. You take any example. I am taking photosynthesis only an example. See, I am giving you the definition to you. What is the definition I am giving to you? At which particular wavelength photosynthesis occur maximum. That is called action spectrum. What friends? At which particular wavelength photosynthesis occurs maximum. That is called action spectra. So you tell me, if I ask you a question, what is the suitable wavelength for the photosynthesis? Every student will give the answer 600 to 700 nanometers is the most suitable wavelength for the process of photosynthesis occurs. You can ask me why. The reason is that all the pigments will absorb maximum amount of the sunlight at this particular wavelength only. What friends? All the pigments absorb maximum amount of the sunlight at this wavelength. That's the reason only 600 to 700 nanometers is the most suitable wavelength. Never get confused with that. So now every student understand what is absorption spectra, what is action spectra. So friends, now the lesson is becoming tougher and tougher. If you understand the next 5 slides very correctly, next 5 slides, entire light reactions will be very clear. If you don't understand the next 5 slides, you never understand the light reactions. Okay, now we all students understand that light reactions are taking place where friends? On the thylakoid membrane. All the students has to follow me. On the thylakoid membrane, every student please understand me. There is a presence of LHC. What is that friends? LHC. Light harvesting complexes are present. Most of the people will get confused here. Every student follow me. This is the thylakoid membrane. Every one of you. This is called LHC. What is that? Light harvesting complex. Inside the LHC, what is present? You only tell me. Inside the LHC, friends, photosystems are present. Say example, photosystem 1, photosystem 2. Now you tell me, inside the photosystems, what are present? That is important for exam. Inside the photosystem, these two are present. What are? Antenna pigments, reaction center. Antenna pigments, reaction center. Every student already know that. What are antenna pigments? Chlorophyll BCD, carotenoid, phycobilin. What are principal pigment? Chlorophyll A. So my question to all of you is very clear. Plant contain chlorophyll A, mostly chlorophyll B in the form of antenna pigments, carotenoid and phycobilin. Never ever get confused with this point. This is only most toughest concept. You have to understand me correctly. If you understand it, it is very easy. So friends, you know very well there are two type of photosystems are there in the higher plants. Photosystem 1 and photosystem 2. I told to all of you many number of times. Why we have given the name photosystem 1 and photosystem 2? I told you many times, based on the order of discovery or the series of discovery. There is no reason behind this. It's very easy. Photosystem 1 discovered first, that's why given the PS1. Photosystem 2 discovered later, that's why they have given PS2. Friends, what is important for the exam? This is important for exam. What reaction center also called as principal pigment. So why PS1 you can call P700? This is the question. Why? Because friends, reaction center contain more chlorophyll A when compared to the chlorophyll B. So in the PS1, more chlorophyll A is present in the reaction center. This chlorophyll A absorb maximum sunlight at 700 nanometers. That is the reason only we have given the name P700. All the old students remember the extra points which we have not discussed. Now, why we call photosystem 2? In the photosystem 2, reaction center is present. Here, equal amount of chlorophyll A is present, equal amount of chlorophyll B is present. Now, this chlorophyll A and chlorophyll B, whichever present in the reaction center, they absorb maximum amount of the sunlight at 680 nanometers. That's why we called P680. So, based on the wavelength absorbed by the reaction center, we called P700, we called P680. And once again, you can get the question directly in the exam. What you can get the question directly in the exam? PS1 and PS2, where they are present? In the plant. Same type of a PS1, same type, which means what? Same P700. It is present in the green sulfur bacteria also. Same PS1. And same PS2, it is present in the purple bacteria also. These are all special points you have to remember compulsory. So see this beautiful diagram. You're all able to understand, friends. This is what membrane, friends? Thylakoid membrane. And this entire thing is called as what? LHC. Inside the LHC, what are present? You only look into this. These are all what? Antenna pigments, accessory pigments. Now, friends, remember, if you understand this slide, 70% of the light reactions are crystal clear. If you don't understand this slide, you never understand the light reactions. So entire light reactions are dependent on this slide. If you get a too difficult question, very, very difficult question, you will expect from here only. So I want you to understand me correctly. Now, never ever forget the goal of the light reactions. What is the goal of light reactions? I want you to solve the questions. Listen. The ultimate goal of the light reactions are production of ATP, production of NADPH. These two are the major functions of the light reactions. How can we do this? Everyone try to understand me correctly. So friends, these are all what? Green color one you can see, right? They are the antenna pigments, also called as what pigments? Accessory pigments. What they made up of? They made up of chlorophyll B, they made up of carotenoid, they made up of xanthophylls. And the dark green color one you can see, no friends, that is what? Reaction center. Reaction center is made up of what, friends? Chlorophyll A. And the purple color you can see, no, that is only we can called electron acceptor complexes. Now all the students follow me. What happens in the plant? How we are going to how the plant produce ATP and NADPH. Try to understand the depth very correctly. Look into this. All the antenna pigments absorb the light, absorb the photon. Look into this. And it is transferring from one antenna pigment to the another antenna pigment. You can see, right? One antenna pigment to the another antenna pigment. My friends, this transfer of energy from one antenna pigment to the another antenna pigment is called as resonance transfer of energy. What do you call, friends? Resonance transfer of energy. So now you people listen. Once it will transfer to, now finally the energy is reaching to where? Energy is reaching to reaction center. Now every student has to follow me. When energy reaches to the reaction center, you know what happens? Reaction center get excited. What, friends? It get excited because it receives a lot of energy. If you eat lot of food, you will get more energy, right? Same. When the reaction center receive more energy, it get excited. When it get excited, it release an excited electron. So friends, that excited electron accepted by several electron accepted, not one or two, there are many electron acceptor, 1 2 3 4 5 6, many. I will tell you. So friends, electron is moving from one electron acceptor to the another. So friends, when the electron is moving from one electron acceptor to the another electron acceptor, some energy will be lost. I'm using the word energy is losing. Friends, that energy only we used to produce ATP. This is how the plant producing the ATP. I will explain you crystal clearly how we produce. You automatically understand. So friends, the question which is asked in the degree level, set level, net level, in your DL level, JL level, listen. What is the movement of electrons? That is the question. Electron always move downhill. Means from one to the other, it is moving. This movement is called downhill movement. Always remember this particular point. And friends, you tell me, this is one reaction center, this is one antenna pigment. First it gain the electron. Gaining electron is called reduction. Later immediately it will send the electron to other, then it lose the electron, then that is called oxidation. So together called as what? Redox reactions. Correct or not? So my friends, my question to every student is, on the antenna pigment, oxidation reduction reactions are taking place. That is very important statement every student has to remember. How oxidation and reduction reactions are taking place? One gain the electron, one lose the electron. One gain the electron, one lose the electron. So that's why we can call it oxidation reduction reactions. Now I will explain you how ATP and NADPH are produced. 80% of your JL and DL, 80%. If they give you the question from the light reactions, 90%, friends, you will get the question from dark reactions only, 90%. But in your JL and DL, you will expect minimum 1 to 2 questions compulsory from the photosynthesis. So they will cover both light as well as the dark reactions. So 80% of the questions will come from here. In the light reactions I am talking, photophosphorylation. Means now I will prove to all of you how ATP produced, how NADPH is produced. So listen to me 10 minutes very correctly. We will solve the questions. Follow me everyone. First one, photophosphorylation. What do you mean by the word phosphorylation? Phosphorylation is a process of joining of ADP with PI to produce ATP. Now you tell me, friends, you only tell me. Now use your brain. You are joining ADP with PI. Yes or no? Yes. If you want to join ADP with PI, what do you require? Tell me. You require energy. Now you have to ask me, sir, from where the energy is coming from? From here. What I told you. When electron is moving from one electron acceptor to the another electron acceptor, it lost the energy. That energy used by the plant to join ADP with PI, then they get energy. So plant actually using the energy to produce the energy. Simple concept it is. So now listen. What kind of questions we will expect in the exam? The type of photophosphorylation. You all people studied in the intermediate level many times. There are two type of phosphorylations are there. Cyclic photophosphorylation, non-cyclic photophosphorylation. I will explain you briefly. Remember, first statement for the exam, cyclic photophosphorylation never ever takes place in the plants. This is the first statement. Cyclic photophosphorylation only takes place in the bacteria. Second point. And in the cyclic photophosphorylation, only one photosystem is involved. Very, very important. And which photosystem? Photosystem 1 only present. 2 is absent. Now every student look into this. If you look into the diagram, you automatically get the question. Every one of you pay attention. Look into this. These are all what, friends? Antenna pigments. The yellow color one is what? Reaction center. You only look into this. Now antenna pigments absorb the light, send the energy to the reaction center. What happens to the reaction center? Reaction center excited now. When the reaction center get excited, the reaction center lost the electron or release the, sorry, it get excited and release the electron. And who is accepting that electron? Electron acceptors. So I told you, right, 90% of the cases you will get the questions from here. So you have to remember, who is the first electron acceptor? Ferredoxin. Second electron acceptor, plastoquinone. There are two type of plastoquinones are there. Plastoquinone A, plastoquinone B. Then cytochrome B6F complex. Then plastocyanin. My friends, listen. So then from the plastocyanin, you only listen, electrons are coming back to where? Electrons are coming back to the reaction center once again. That is the reason we can called cyclic photophosphorylation. You know, I think someone asked me question. Anusha, I think you have asked, right? In the NEET 2020 question paper also, this is the only question asked. Plastoquinone transfer the electron between FD to cytochrome B6F. This is the question asked for the 4 marker. Very easy question. And of course, you will get that kind of questions only, undoubtedly. Okay, now listen. This is in which cyclic photophosphorylation? All the students, you have to think and answer my questions. Here, electron is moving from FD to PQ, PQ to cytochrome B6F, cytochrome B6F to the plastocyanin. Every student follow my words. Now, friends, when the electron is moving from one to the other, it lose energy. See, friends, electron always lose energy. That energy only used to join ADP with PI. Then we get ATP. Now, every student, please answer my question. My question is very clear. Do we fulfilled the goal of light reactions by using cyclic photophosphorylation or not? That is my question. Do we fulfilled the goal of light reactions by using cyclic photophosphorylation or not? Of course not. What is the goal of light reactions? The goal of light reaction is production of ATP also, production of NADPH also. There are more than 5 times the same question asked in the NEET also, the same question asked in the set also. What is the question asked? In which of the following phosphorylation reducing equivalent is not produced? This is the question asked. What is the question asked? In which of the following photophosphorylation reducing equivalent is not produced? All the students remember my words. That is the cyclic photophosphorylation. We did not produce NADPH. What is the answer? Why? What is the reason? The reason is only one photosystem, one is present. Only one photosystem is present. That's the reason. Never ever get confused with that. Okay. Cyclic photophosphorylation is happening where? I told you, in the bacteria. Most important one, the very, very important one, that is called non-cyclic photophosphorylation. Every student, please listen to me very correctly without confusion. Non-cyclic photophosphorylation, how many photosystems are there? This is utmost important, friends. Very important. 2 photosystems are involved. And most importantly, where it will occur? It occurs in the higher plants. Now you only look into this. Why did I called non-cyclic and why did I call cyclic? Because electron movement started here. But friends, electron reached where? It reached same position, right? That's why we can call cyclic. But you tell me, electron started here. Does the electron is coming back to this place? Never ever. That's why we can called non-cyclic photophosphorylation. You only look into the picture. In case of a non-cyclic photophosphorylation, how the electrons are moving. Look into this. Electrons are moving in the form of a sleeping jet, like this. That is the reason non-cyclic photophosphorylation, also called as JED scheme, also called as zigzag scheme. Very important point. Now look into this beautiful thing. See, once again, every student will give the wrong answer. They will always compare electron flow between the cyclic and non-cyclic. You should not do that, because cyclic is happening in the bacteria, non-cyclic is happening in the plant. How can you compare? You should not compare. Okay, that's why most of the people will make it wrong. But just to remember for easy understanding, in the non-cyclic, what is the first electron acceptor? Ferredoxin. After that, plastoquinone. After that, cytochrome B6F. After that, plastocyanin. This is in the cyclic. Now every student follow my words. In the non-cyclic, this is very interesting one. In the non-cyclic, you can produce NADPH also. You can produce ATP also, no doubt about it. Now listen all the students, very important and very interesting and very difficult. Now listen, all the students listen. Now first P680 absorb the light and send the electrons to the reaction center. Every student remember. And from the reaction center, who is accepting the electron? The first electron acceptor called pheophytin. Every student remember, this is a very important one. Pheophytin. After that, quinone A. Quinone B, same, no same. Quinone, cytochrome B6, plastocyanin. In case of cyclic, first electron acceptor is ferredoxin. In case of non-cyclic, it is pheophytin. That's all. Quinone A, quinone B, cytochrome B6F, and plastocyanin. Correct, na? Friends, listen. Plastocyanin, then electron moving to where? You only tell me. Electron moves to P700. From P700 to reaction center. From reaction center, who is accepting the electron? A naught is accepting electron. My friends, every student has to listen my statements. A naught also called as modified chlorophyll A. Never ever forget this point. I will ask you in the tomorrow class. A naught is also called modified chlorophyll A. A1, it is called as a phylloquinone. It is also a pigment. Phylloquinone, followed by iron sulfur X, iron sulfur A, iron sulfur B, ferredoxin, FNR. FNR means ferredoxin NADP reductase enzyme. Then followed by NADP plus, then followed by NADPH. In this particular diagram, I have not shown you ATP production. I will show you in the ATP production in the next slide. But in the non-cyclic, we produce NADPH also. We produce ATP also. Okay. Now all the students, once again, I will explain. Let me ask you a simple question. Every student listen. So from the P680, electron accepted by pheophytin, you have to answer the questions. Be ready. Quinone A, quinone B, cytochrome B6F, plastocyanin. Then from the P700, A0, A1, series of iron sulfur proteins, FD, FNR, NADP plus. Now friends, my simple question to every student is, who is the electron? This is also direct asked question. Who is the electron donor for P700? Look the diagram and tell me. Who is the electron donor for the P700? Who is the electron donor for the P700? Every student, why you are thinking? What is there to think? Electron donor is what? PC. No, 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 no. Gayatri, you should not say PS1. It is what? No. How it is water molecule? What you people are looking at? Morally look into the question correctly. What did I ask you a question? Who is the electron donor for the P700? Who is donating electron? Plastocyanin is donating. Correct na? Now every student answer my question. Who is the first electron acceptor? Remember everyone. Who is the first electron acceptor from P700? Who is the first electron acceptor from P700? Who is the first electron acceptor from P700? Every student. FDR. Who is the electron acceptor from the P700? Which is the A0. Correct, na? This is what? A0. Not to confuse. It's easy, right? Very good. Very good. So friends, now I am going to tell you the most very interesting topic which we did not read last time. Okay. Now all the students listen. Most of the people will think that first photosystem 2 will work. Later photosystem 1 will work. That is wrong statement, friends. Every student remember for the exam. Photosystem 1 also absorbing the light continuously. Photosystem 2 also absorbing the light continuously. Both work simultaneously. They can give this statement only in the exam. They will give the statement that all two photosystem work in a series, which is wrong statement. Two photosystem work simultaneously. That's an important statement. Now every student listen, not to confuse. P700, every student answer. P700 absorb the light and send the electrons to the reaction center. So friends, P700 now it is deficient of electron because it send the electron to reaction center. Now, who is donating that particular deficient electron? Who will donate? Plastocyanin will donate that deficient electron. Okay, you got it. Very good. Similarly, P680 also absorbing the light. P680 also transfer the electrons to the reaction center. Now P680 also deficient of electron. Every student remember. Now who is the electron donor for the P680? Every student quickly answer. Who is the electron donor for the P680? Who is donating? Water molecule. So now Varalakshmi, now look into this. The question which you have asked last time, look into this. So friends, breakdown of water, we call it as what, friends? Photolysis of water. Follow me, every student follow me. Last time I did not explain to you this. Photolysis of water. What is photolysis? Photo, in the presence of light. Lysis, breakdown. What is breaking down? Water is breaking down. When you break down the water, you get 2 protons, you get 2 electrons, you get a half oxygen molecule. My friends, such an important question here. You all people know that this is only called oxygen evolving complex or called as water splitting complex. Every student know that in order to split the water molecule, this complex require, what they require? 4 manganese, and every student remember they require 4 chlorine, they require 4 calcium. All are required, friends. Never get confused with that. Don't think that only there is a requirement of the manganese. There is a requirement of chlorine also. There is a requirement of the calcium also. Now, what is the very interesting question which you have never read? This is called photolysis. Now you tell me, who required the electron? Tell me, friends. P680 required electron. Yes or no? Of course, yes. My friends, you are all able to see in the purple color, right? Purple color. That is only called as a oxygen evolving complex. You also called as water splitting complex. It only contain manganese, it only contain chlorine, it only contain calcium. My friends, what is my question to everyone? Okay, when you break down a water, this is the most important one. When you break down a water, you get 2 proton, you get 2 electron, you get oxygen. My friends, you tell me where the proton released out? Proton released into the lumen. Where the oxygen goes? Oxygen, this is the oxygen what we are breathing. This is the one which goes into the cellular respiration. My friends, now the question is here, who is accepting electron? That is the question. Who will accept electron? You only look into this. P680 is accepting the electron. Now, what is my interesting question to all of you? Inside the P680, inside the P680, there is a protein is present. What is the name of the protein? Protein called D1 protein. That D1 protein will up. So that is also question, friends. That is also question. Which protein of the P680 absorb the electron. That is the question, which is called D1 protein. D1 protein absorb the electron. Simple statement. Now you understand? Varalakshmi, is it clear? How does the water molecule break down? Friends, now Jyoti, you remember you have asked a Q cycle, is it? So now focus on the Q cycle. I will explain. Okay, now focus. What is Q cycle? Now friends, in this particular diagram, I have explained you NADPH. Now I have to explain you how the ATP get produced. 90% of the teachers won't explain how the ATP get be synthesized. They will just say that proton motive force. But how does this particular proton motive force will be formed? That's a very good question. Very important question it is. Now, friends, in order to explain the ATP synthesis, there is a theory was proposed. What is that theory? We can call it chemiosmotic theory, proposed by a physiologist called as Peter Mitchell. You know, I don't explain about the name of the scientist. You have to remember it. Okay. Not to confuse my friends, I will explain you in 10 minutes. Very briefly, I will end the class because this is a very interesting concept. How the ATP get be synthesized. Focus everyone. So tell me once again, remember we are synthesizing the ATP friends via proton motive force. Remember this point. Now all the students follow me, all the students. See friends, keep remembering my words. From the P680, who is accepting the electron? Pheophytin is accepting the electron. Yes or no? Yes. From the pheophytin, who is accepting the electron? Plastoquinone is accepting the electron. Yes or no? Yes. Now remember, Jyoti, listen to me very correctly. What is Q cycle? Okay. See friends, now plastoquinone, every student follow me. Plastoquinone will accept electron from the pheophytin. When the plastoquinone accept the electron, PQ can reduce to PQH2. What friends? PQ reduced to PQH2. Never forget. But friends, that is not the question. If the PQ want to reduce to the PQH2, not only electron, it also require proton. My friends, that's a very important statement. It also requires the proton. Now you tell me from where the proton is coming. Proton is coming from stroma. Every student follow me. No need to confuse. It's very easy. Plastoquinone. Very good. Plastoquinone accept the electron from pheophytin, accept the proton from the stroma, and plastoquinone reduced to PQH2. What friends? Plastoquinone reduced to PQH2. Now you tell me, do you think PQH2 will keep the electron with it? No. It has to send the electron compulsory. Compulsory to where? Compulsory to the cytochrome B6F complex. Now friends, you have to understand me. Now PQH2, once again it undergo the oxidation. So what do you mean by that? It is sending the electron to the cytochrome B6F. Then PQH2 come back to the PQ again. So friends, okay, it send the electron to the cytochrome B6F. Now tell me, what about the proton? That is the question. Proton released into the lumen. You can see that proton is released into the lumen. So friends, when the proton is released into the lumen, and just now we studied, when water molecule break down, that time also protons are released. So friends, now proton concentration is becoming more and more and more and more in the lumen when compared to the stroma. Once again, I'm explaining you, all the students listen. I'm explaining you how the ATP synthesis can be occurred. Plastoquinone accept electron from the pheophytin, accept the proton from the stroma. How many electron? 2 electron, 2 proton. Very important. Then PQ reduced to PQH2. Then PQH2 immediately send the electron to cytochrome B6F complex. Then PQH2 oxidized back. So friends, plastoquinone undergo the reduction. PQ to PQH2. And PQH2 it come back to the PQ, that is oxidation. So every time it is sending electron to cytochrome B6F, it release the protons into the lumen. So this oxidation and reduction carried by the plastoquinone, we can call Q cycle. But in the biochemistry, we will read little elaboratively, but you try to understand here also. Now remember everyone. So tell me, finally, where the proton concentration became more? Proton concentration became more. Where? In the lumen. So friends, now what we have to do? Now we have to maintain the charge balance. Where? In the stroma. So now you know what happened. Proton is moving. From where to where? You only look into this. Proton is moving from the lumen into the stroma. Friends, how the proton is moving from the lumen into the stroma? Via an important enzyme complex we can call the ATP synthase complex. You can also call what complex? F0 F1 complex. Correct na? So when the proton is moving in the form of a wave like this, every student remember, proton always generates energy. So friends, when the proton generates energy, now proton came out. Now that energy is used to join the ADP with the PI. Hence, there is a production of ATP molecule. This is how we can produce ATP. This is called proton motive force. So now I am asking you a very simple question to all of you now. Where does the photolysis of water takes place? Is it taking place in the cyclic photophosphorylation or is it taking place in the non-cyclic? Every student. Wonderful. Only in the non-cyclic photophosphorylation only it is taking place. Never. And every student answer. Where the oxygen is released, is it released in the cyclic photophosphorylation or in the non-cyclic? I am talking about release of oxygen. Every one of you. Of course, friends, release of oxygen also non-cyclic only, right? Wherever the water breakdown, there only oxygen will be released. Now, every student answer my question. Every student answer my question. How many number of protons, how many number of electrons, how many number of oxygen molecules are released when 100 water molecules are breakdown? This is my question. How many number of protons, how many number of electrons, how many number of oxygens will be released when we break down 100 water molecules? Every student has to answer. How many number of protons, how many number of electrons, how many number of oxygen molecules are released when 100 water molecules are breakdown? Always remember 200 proton, 200 electron, half. Because friends, one water molecule always give, no. Someone said 100, who said 100? Look into this, Jyoti. One water molecule always give half oxygen. Not to confuse. It will always give half oxygen. So half oxygen means what? You get only, for 100 water molecule, you will get only 50 oxygen. 200 protons and 200 electrons. Not to get confused, not to get confused. Good, very good. So friends, I hope everyone understand the cyclic and non-cyclic very elaboratively. I strongly believe. Okay. Okay, now I am showing you one very important question. I want every student to answer. If you answer this, I believe that you are getting me correctly. Every student has to answer this. Every one of you, all 44 people has to answer. Don't sit silently in the class. I'm just saying to all of you, think and answer, not to hurry. I want everyone to answer, but think it, don't be hurry. I am expecting everyone will give the right answer. Wonderful. Others, everyone. Don't sleep. B. Gayatri, why B, Gayatri? You know, friends, very good. Answer is D. You know, this is the CSR net question which is asked in the 2022 September. See here. Now you all people answered CSR question also just like that, very, very easily. Right? Very good. Why option A is wrong? So first P682 pheophytin. From the pheophytin to plastoquinone. But option A says that electron from P680 moves first to quinone. This is wrong statement, right? P700 can receive electron from plastocyanin, right statement. NADPH is produced at the end of light reaction, right statement. Hydrogen ions are the protons. During light reaction get concentrated in the lumen. Of course, right statement. The correct answer is D. Wonderful, amazing. Understand Gayatri correctly? Okay, very good, very good, very good. Quickly answer this question, question number 73. Every one of you. Wonderful. Okay, remaining questions we will solve in the tomorrow class. C. How C, man? See, you can see, right? Why people are thinking this much of time? It's easy question, right? The last electron accepted is obviously plastocyanin. No, you can easily answer the question. P680, plastoquinone, cytochrome B6F, then plastocyanin. Anyway, they did not given the pheophytin. Correct, na? Hence, the correct answer is obviously the D. Okay, remaining questions we will discuss in the tomorrow class. Okay, everyone say yes or no. Is it clear, everyone, so far? And please let me know, especially the new people, the speed is sufficient for you or you want me to go little slow? The new students. Is it clear? The speed is okay for you? I hope I am not going too fast. Clear, everyone? Others, new people also. Okay, now all the students listen. Okay. Once again, I am telling to every one of you friends, remember my words. If you don't study consistently for 100 days, you never ever crack the exam. Reading 1 or 2 days is not important, friends. You have to read 100 days. You know, 99.99%, just now I have seen the newspaper, the Gurukula board is also going to complete the exam within the September only. So you are having both exams on the same time. So these 100 days are very crucial. I'm requesting you read 5 to 6 hours minimum compulsory. Otherwise you can't crack the exam. So now listen, you are already having a photosynthesis PDF. Once again, I'll send you in the WhatsApp group right now. So listen, where every student is failing is you are not reading, you're just reading the lesson. That's all. So now keep the PPT with you and read the photosynthesis PDF complete light reactions by tomorrow class. By that you will get a wonderful idea. In that PDF only there are 300 practice questions are there. You can do the practice questions once you complete the dark reactions also. But without reading, don't attend the tomorrow class. Okay, keep the PPT and read the information. It will become very, very easy to all of you, but read compulsory. Okay, I will see you in the evening 5 p.m. in the cell biology class. Okay. Okay, friends. Thank you. Thank you all. Thank you for your patience. I'll see you in the tomorrow class. Keep reading. Okay. Bye-bye. Thank you all. That's great.